Okay, I guess we've reached the 1 o'clock hour. I guess we should start off with a few announcements. I guess probably the most pertinent would be the exam that everyone took. So just a quick announcement, and I don't know if Dr. Parker is going to want to jump in here, but we have awarded another point back to everyone. And Keith, do you remember what the question number was for that?
It's question 15.
Okay, so do you want to say anything about that at all, or?
No, it just looked to me like the numbers were a little bit low, and I thought it would just fit better if we gave credit for it.
Okay, great. So if you check out your grades, they've been updated in Moodle already. And so you should reflect that additional point back to everyone who took the exam, with the exception of those folks who kind of hit that maximum 50 points. Obviously, that's not going to affect you. In terms of getting your physical exams back to see kind of how you did, I'll be delivering those later this afternoon. We just had a few people who had to take their exams at DSS, and I just got those back yesterday. So I'll be updating those grades and then getting the physical copies of the exams back to you this afternoon. I'll be putting them in your mailboxes over in the, I guess, northern part of Skaggs. So I guess to kick off, does anybody have any questions about that at all? Okay. And I guess everyone has the pleasure today of getting a essentially hybrid lecture. So I'm going to finish up a few slides I have remaining on the endocannabinoid system, and then we're going to kick it on over to Dr. Parker, and he's going to finish up today's lecture. So you kind of get to have the experience of both of us in one day. So hopefully that's a kind of a fun experience for everyone. So yeah, if no one has any questions or concerns, please feel free as I'm lecturing to pop any questions you might have up in the chat. And if you have any questions about the existing material or previous material, I'm happy to answer those questions. I'm going to be sticking around for all of the lectures and recording and participating anyway. So again, if at any point something comes up that you'd like to talk about, I'm more than happy to do so. And I see Keith, so I'm going to say, what's up?
I just want to say, take your time to do whatever you need with your material, because I'm just going to be fooling around with a few little things afterwards, which are fine, but you've got a really important segment there. So don't be shy at all about taking as much time as you want, or having as much interchange between yourself and the students as necessary, because it's a topic that I think is a worthy one for discussion. So anyway, there we go.
Okay, so let's go ahead and get started with today's lecture then. Okay, so today's lecture, again, we're just going to be finishing up some material on the endocannabinoids. So I think in the title of this, it's cannabinoids. I like to say endocannabinoids because often we're talking about the endogenous receptors when we're talking about these different neuromodulatory systems. So when we talk about dopamine, we're talking about the dopamine receptors. We're not necessarily talking about, L-Dopa or other types of exogenous things that are activating these receptors. And so if you're following along in the textbook, most of the material that I'm lecturing on today was taken from Goodman and Gilman as well as Katzum. I will say that the chapters surrounding this particular topic are pretty sparse at this point in time. I did dig into a couple of reviews to pull out some additional material that I think is relevant, just because this is such a rapidly evolving field. And I think the research is really just starting to get started here in terms of the therapeutic I guess, approach that we're using for cannabinoids. I think that the textbook just kind of hasn't caught up to where the field currently is. And I think it's going to be an area of therapeutics we're just going to see expand over time. And, I'll allude to that a couple different times within this lecture. But I see Keith, and I feel like you definitely have a opinion about this.
Yeah, just to comment on the books again, it's a real shame, really, that pharmacology textbooks have been so lethargic in addressing topics associated with cannabis. And you're absolutely right, Katie. The coverage is, incredibly light on this very important topic. And
Mm-hmm.
I certainly hope that very, very soon in new editions of these textbooks, they correct that. that problem, because there's really no excuse for such light coverage that they've done.
Yeah,
Okay.
and it's also kind of, it's interesting that they get characterized in drugs of abuse, as well. Those are some of the chapters that you're seeing them categorized in here, instead of looking at them as more of a therapeutic. They're, I think, thinking of them more in the recreational perspective. Okay, so what have we discussed thus far, and what are we going to be talking about today? So we've talked a little bit about what cannabinoids do, some of the chemistry, we'll do a quick review of the synaptic properties and the receptor properties, and we hit upon a few figures that show distribution in the body. So today we'll wrap up talking about some physiological properties, disease states, therapeutic approaches, as well as ADME. So just a quick review as to what we covered last week. I know sometimes, you hit that 4-day week and you kind of forget exactly where we left off. So again, the endogenous cannabinoid system and kind of the receptor expression is a little different than a lot of the other synaptic receptors we've talked about, just because this is very much a retrograde signaling system. And so you can see here, instead of, you're seeing postsynaptic, presynaptic release of neurotransmitter. In this instance, you're seeing postsynaptic release of the signaling molecules, things like anandamide as well as 2-AG. Those are going to be released through the anandamide transporter and then essentially activate those presynaptic CB receptors. We'll talk about both receptor subtypes, CB1 and CB2. And since we're talking about it here in terms of, neurophysiology, this is most likely the CB1 receptor that we're looking at here. You can see here that it's a G protein coupled receptor. It has 7 transmembrane domains. It's often coupled to G alpha I, G alpha O. And you can see some of the downstream signaling pathways that get activated with that. Generally, what occurs upon release of cannabinoids and activation of these receptors is that you see a decrease in presynaptic release. And so generally, it has almost a dampening down effect of whatever system that you're looking at. So when we talk about inhibitory neurotransmitters, this isn't unlike that in the fact that you're inhibiting presynaptic release of the neurotransmitter. And so here you can see by that red line how it's blocking exocytosis and blocking those voltage-gated calcium channels. So those are going to be kind of the downstream signaling effects of activation of the CB receptor. And so it's interesting to think again about where the receptor is distributed and kind of the downstream effects that receptor is going to have at that specific location. I guess the only other thing that's kind of worth mentioning here is that the postsynaptic kind of metabolism and generation of anandamide is shown here at the bottom of this particular figure. And you can see kind of there's an excessive stimuli of postsynaptic receptors that causes endocannabinoid synthesis. And so just speaking from my own experience, I can say that there are specific muscarinic receptor subtypes that lead to elevated levels of essentially downstream signaling cascades that lead to endocannabinoid synthesis. And so again, this is very much still a very hot topic of research in terms of understanding the whole cycle here. And it kind of, as Keith and I are continually alluding to, I think for a long time, this area of research was really neglected. And now is kind of seeing its heyday, I think, again. In terms of looking at receptor distribution, this is a slightly different figure than what I showed you before, but I like this because it not only shows you distribution, but then also talks about what physiological effects it has in those regions. And so we'll hit upon this later when we talk about physiological effects, but I like this figure because it introduces a lot of those ideas. And you can see that both CB1 and CB2 are expressed in the brain. CB1 maybe has a little bit more effect on things like cognition. We're talking about cognitive deficits that occur that's most often mediated by CB1. When you think about CB1 and CB2, in my own mind, when I'm characterizing them, I always think of CB1 as being far more cognitively related, and then CB2 being far more immune cell kind of regulated. There are some exceptions to that rule, as with any rule, but those are kind of the broad generalizations I generally think of these things. You can see again that they're expressed in the brain, in the heart, in the liver, spleen, the GI tract, as well as skeletal muscles and bones. So again, I like this figure because it's a great synopsis of showing you both receptor expression and some of the physiological essentially pathways that these receptors might affect downstream. So again, it's not a receptor that is solely expressed in one region. It's kind of pervasive across body and can have various either activating or deactivating relationship with those processes. Okay, so when we talk about the cannabinoid system, it's hard not to kind of kick off that conversation talking about the marijuana plant, just because I think historically that's often the way that this particular receptor system came about in terms of understanding what exactly it does. Because of the kind of co-evolution of humans and the medicinal practices and uses of the marijuana plant, often we think about their relationship being very closely tied. What's interesting to note, though, is that when we think about the marijuana plant, we most often probably think of that THC product that has those cognitive effects that people are kind of seeking out the use of the marijuana plant for. But it does have other medicinal uses, as you know, historically we've seen throughout the evolution of man. So here is a nice little kind of laundry list of different, essentially agonists of the CBD receptor that have been isolated from the marijuana plant. So this is again kind of a whole naturopathic area of research that's still is still quite a hot topic. But when we talk, but currently, I would say the two kind of therapeutic compounds that have been distilled from the marijuana plant are probably THC and then cannabidiol or CBD. And so I showed this figure last time. I think it does a nice job of comparing and contrasting some of the different compounds and talking about, again, how some of them are more psychoactive and some of them are non-psychoactive. And again, often the relationship between them being psychoactive and non-psychoactive is a direct, I guess, response or readout of what types of receptors that they're interacting with. So not surprisingly, THC, CBN, THCV are going to be more of those psychoactive components that are interacting with CB1, and then CBD, CBG, and CBC are compounds that are more likely interacting with CB2 preferentially. So again, those different receptor expressions and the affinity of these compounds for those receptors is going to essentially alter the physiological response. So I kind of like breaking down again. I'm going to focus primarily on THC and CBD, just because of the two compounds I think that we have the most information about at this point, and has been, you know, most information and has been the most investigated. And like I kind of keep alluding to the fact that these different agonists have different preferences for those receptors. And so if we compare and contrast THC with CBD, THC is a partial agonist for CB1 and CB2, while CBD is an agonist for CB2 and may bind to other receptors. And so that's still, again, an area of hot research. And I see Keith's face, so I'm excited to see what he has to say.
I just want to add in that there's also a lot of non-cannabinoids in cannabis. There are like terpenes, for example, which are relatively quote unquote simple carbon hydrogen type compounds. And with the emphasis over the years of understanding what's going on with THC, these other compounds as well as CBD have been pretty much ignored. And that isn't to say there hasn't been a lot of research done on cannabis. There has been. It's one of the misnomers when you see some of the popular TV programs and so on. They say, well, we really haven't studied cannabis very much. That is actually no more true of cannabis than a lot of other drugs out there. But I just wanted to jump in and say that it'll be interesting over everyone's lifetimes to see what we discover about some of these other non-cannabinoidal substances in marijuana, and what role they all play in the big picture. So okay, there we go.
Yeah, it was interesting too, as I started to dig into reviews, how there are other agonists for the cannabinoid receptors that obviously exist outside of the marijuana plant. You know, there are different types of animal toxins and things like that that show a certain amount of affinity for the cannabinoids. So I think we're still also, you know, figuring out other types of maybe natural products or even synthetic products that show an affinity for this receptor class.
I was wondering, have there been any antagonists found?
Oh, natural antagonists. So if we look at that CBD, you can see here it actually has an antagonist or inverse agonist effect on CB1. And so CBD can actually have that effect on the CB1 receptor. And so there's some natural products that have been found to be antagonists, but there's also been a lot of synthetic compounds that have been generated as well. Again, just because this isn't a hot area of research, and as you're doing the pharmacological characterization of any receptor, it's important that you have kind of both of those tools in your toolkit. And so there are synthetic products that act as agonists as well. And there have been some antagonists that have been used therapeutically, but I don't know if we'll discuss those today. Does that answer your question?
Yeah, thank you.
Okay. Sometimes I start answering a question, I just want to make sure I don't, get too far off track. Okay, so kind of coming back to THC and CBD. Again, THC, because it does have those cognitive effects, it's more the high that people generally describe, and are going to alter things like perception, reaction time, learning, and memory. And those are going to be impaired for about 2 hours. We'll talk about the ADME of some of these products. Tracking coordination may be effective for several hours, increased giddiness and increased hunger. Again, because of this increased hunger, this THC has been used as a therapeutic to stimulate appetite. We'll talk about that. Some negative side effects include panic, hallucinations, acute psychosis, more often seen when THC is ingested and not necessarily smoked. And I think there was a short discussion, I don't know if it was in the textbook or in a review, about this psychosis that occurs upon THC ingestion, whether it increases or it occurs at a higher rate in those folks who kind of have a predisposition for schizophrenia, and that's kind of what leads to that psychotic or psychosis experience. CBD again is considered, I think, a little bit more therapeutically relevant, especially when we're talking about kind of the peripheral body, because it acts as that anti-inflammatory, analgesic, anti-anxiety, and anti-tumor properties. And so it kind of has a separate set of receptor activation downstream pathways that are getting activated. Kind of an advantage to CBD is it does have a low addictive, hallucinogenic, and toxic side effect kind of profile. And then the first approved CBD kind of activation, the Epidiolex, was used to treat rare and severe forms of epilepsy. I included this table here just because I think it does a nice job, again, of outlining these two different chemicals that we're talking about in terms of exactly how they act pharmacologically, and then getting more into their receptor kinetics, kind of their EC and IC50s, as well as kind of their bioactivities and what they're used for therapeutically, which we'll talk more about here in the subsequent slides. But I think that this slide does a nice job of comparing and contrasting THC and CBD in several different ways. Okay, so therapeutic uses. So as we're having this discussion, it's obviously very relevant and fairly timely. I kind of just didn't know exactly where to kind of throw in this this kind of tidbit, is that it is on the ballot for legalization in Montana this November. In terms of therapeutics, has been used and prescribed an antiemetic. So essentially these drugs have been prescribed to combat the side effects that are seen with chemotherapy, whether they be pain or decrease in appetite. It's also been medicinal. Marijuana has been used to combat appetite stimulation in AIDS patients as well. And so here you can see some of the generic names of a few of those drugs, and kind of, again, giving you an idea of exactly what receptors are activating. So this Nebilone is a synthetic CB1, CB2 receptor agonist, and Sativix contains mainly the THC and cannabidiol. You can see other kind of effects here, muscle relaxing effects. You saw, I think on the previous slide, how it's been used for things like multiple sclerosis, anticonvulsant properties. This particular therapeutic use is kind of what led me down my particular area of research, because I was interested in how to mitigate seizures in people who had been exposed to nerve agent. And so looking at the literature and seeing how some of the pathways that are activated with seizures and with epilepsies, how this can is a proven therapeutic. I started to investigate this as a therapeutic for nerve agent induced seizure, which was kind of cool. It obviously has been used to treat glaucoma, and then also as an appetite stimulant. And there was a product that was out there when you asked about antagonists. There was a product that had been used, was out there as an antagonist for the system, as an anti-obesity drug, but has since been removed from the market and is no longer available. So kind of playing with that receptor system, an agonist kind of stimulating appetite, an antagonist essentially being a treatment for obesity.
So sorry, for the appetite stimulant, would that be used like for elderly people who don't have a high appetite to encourage them to eat? Or is it used more to treat like eating disorders or?
I think again that kind of overlaps with that. Folks who are undergoing chemotherapy might have a hard time eating. Anybody who's kind of has a chronic disease that maybe isn't getting enough calories to fight whatever chronic disease they have. But I see Keith's face, so maybe he has something to add to this conversation.
Well, I was just thinking about the conversation everyone was having there relative to metabolic regulation. Uh, it's kind of logical, I guess, in a sense that if marijuana itself is an appetite stimulant, that you could uh, dampen down appetite and and maybe have a little metabolic regulation using a cannabinoidal antagonist, and that's what you were talking about there, and that was marketed by a British pharmaceutical firm, but was removed from the market, not because it didn't work in the metabolic sense, but because it's had some negative effects on with respect to producing depression in susceptible individuals, which also kind of makes sense. And I'm not sure why in the regulatory process for getting the approval that didn't come out, but it didn't, and so unfortunately that was a failure. But it's still an area that's being pursued because, as we all know, the ability to find appropriate metabolic regulators is a very big area of need and a very difficult one to crack. Thank you.
So Keith, before you
Yeah.
to video, in terms of it being used as an appetite stimulant, has that been used generally as a co-therapy for other disease states, or have you heard of it being used in, like I think Rebecca was asking about it, like in the elderly population?
I think both of the above, and I think that at least in the United States, the big problem has been, the regulatory legal one, as opposed to the appropriateness of use in some of those situations that you're talking about. So is that regulatory mess is what it is, gets hopefully untangled over the next few years. Perhaps some people who could really utilize these substances in the realm of metabolism will be able to do so without having to break the law, if you will, in the process. How's that?
No, it's good. It's I've always liked the conversation we get when we get going. So I felt like, if we're going to treat this kind of like any other drug, we should also talk about some of the pharmacokinetics related to it. So there are several different ways in which it can be administered. Obviously, it can be inhaled. There's an oral mucosal route, as well as ingested. It does have a fairly low bioavailability. As once it's been ingested, it distributes very quickly into the well-vascularized organs. So things like the brain, heart, and lungs. Chronic use may lead to accumulation in adipose tissue. So that's something that I didn't necessarily recognize until I was kind of reading about it, is how it can essentially, because it's so lipophilic, it can kind of sit in your adipose tissue. And then, during periods of extreme weight loss, that actually can be released back into the system. In terms of metabolism, it's predominantly hepatic. Both THC and CBD are both metabolized in the liver via cytochrome P450 enzymes. They have specific subtypes of enzymes. If you're interested, I'd be happy to share a review article that was focused on the pharmacokinetics and pharmacodynamics of the cannabinoids. It is excreted in the urine, and the half-life can range pretty widely from 6 minutes to 24 hours, dependence on frequency of use. Again, if you're a chronic user, you have a higher chance of it being distributed in that adipose tissue, and then so that longer kind of release can affect essentially that time to excretion. So, and I guess in terms of because we talk about this as a drug of abuse, that's the chapter that it's characterized in, there is some type of dependence that can develop over time. Again, this is for THC, not necessarily CBD. Again, that being one of the advantages to CBD is that you don't see the type of addictive, I guess, property. And because of that, you also wouldn't see the side effects for withdrawal that we're talking about here. Things like restlessness, irritability, mild agitation, insomnia, nausea, and cramping. So again, these are some of the withdrawal symptoms that would occur upon discontinuation of use of THC. So essentially, that's kind of how I'm wrapping up kind of the cannabinoid kind of section here. But I'd be happy to kind of have a longer discussion about any of this with the students. I didn't create a concept check again because I was trying to keep this short because I realized we would be splitting our time, Keith. But I guess if there's any discussion anyone would like to have about this, I guess it's kind of an open forum at this point, whether you'd like to throw up a question in the chat or even just unmic and voice a thought.
While people are thinking about that, let me jump in and try to not be too excessive in discussion here, but a little bit. I think, it is a, extraordinarily interesting area of societal interest, and my guess is that there probably are questions or topics of discussion out there that people would like to pursue, but maybe haven't jumped in here yet. So let me say a few things and see if we trigger any other discussion in the process. I did note that, Katie, that you mentioned that the upcoming election has relevance with respect to this particular topic, and that's absolutely true. Maybe some people have voted already, and in those cases, you may well be aware of these ballot initiatives that exist on the Montana ballot and in many other states also. So I guess we're just exactly 2 weeks away from the election today. And I would encourage people to, if you are already registered as a voter, to take a look at the information pamphlet that's provided for Montana voters. And it's certainly not my position to suggest to people how they should vote, but I think that it is within our purview to encourage people to vote, because citizenry is something that's a product of all the courses and so on that we take in our education. So to be a citizen and vote, I think is a very, very powerful thing. You will find that in the voter information pamphlet, amid some other things that they talk about there, that there are two ballot initiatives on the Montana ballot. One of them basically has to do with the giving the legislature power to assign an age related thing with respect to cannabis. And the other one, which is a much longer one to read about in the pamphlet, has to do with setting up the commercial enterprise for cannabis in the state. So you folks have lots to do, and it's hard to find time, but if you could spend a few minutes taking a look at that voter information pamphlet, I think you would find it worthwhile. I have some other things that I wanted to say about this matter, so let me see if I can collect my thoughts and pull those out of memory. Did you have something to say, Katie?
I did. I actually had a question. Somebody popped up a question in the chat that I think you might have specific expertise in answering, because they're asking about the depressant effects upon withdrawal. And I looked at some of the previous years and how cannabinoids was and how you presented cannabinoids. And I know that you had talked about some research that you had done with the serotonin receptor. So I wanted to ask you if, with the withdrawal symptoms, could that also be a result of the interaction of the cannabinoids with the 5-HT receptor?
It certainly could, and I'm glad you brought that up. And I did notice in your presentation that you talked about the possibility of other receptor systems being involved. And that's an area that is sort of interestingly exploded in terms of research interest over the last 15 years or so. And cannabidiol, for example, clearly does interact with the 5-HT1A receptor, as well as possibly other serotonin receptors. Studies from just the test tube type study clear through humans and experimental animals and so on uh, support the idea that cannabidiol interacts not only with cannabinoid receptors, but with serotonin receptors, and now with other types of receptors. There's a rather wide range of receptors that seem to be potentially under the influence of cannabidiol. How important all these other receptor systems are is yet to be determined, but I think that the serotonergic ones, there's enough. There's hundreds of references out there now in that particular area to suggest actual clinical relevance of some of these other types of interactions. So I think that's an area to really stay in tune with, and I think it also demonstrates, broad pharmacological principles that we've been talking about, and having to do with classification. I mean, it makes all the sense in the world to think that a cannabinoid like cannabidiol interacts with C, CB receptors. But we don't want to get too stuck on that, because if there are, if by classifying CBD as a cannabinoid and its interactions with cannabidiol receptors is sort of the thing to think about with respect to actions, we may be missing out on a lot of other things. And this is true not only in cannabis, but in really a variety of other pharmacological areas. The classification is powerful. When we think about cannabis, and when we think about cannabinoid receptors, then to emphasize that classification triggers certain ideas in our minds which are fruitful, but it can also be overdone. And so I think it's going to be very, very interesting to see how this plays out over the next few years in terms of in the cannabis area, how much of the overall effects are due to interactions with cannabinoid receptors, and how much are due to interactions with other types of receptors. And we could think about similar sorts of relationships like that with respect to a variety of other drug substances. So I think this is a really sort of neat specific example, but it's also a broadly based example for all of us to think about. I think in terms of adverse effects, well, let me back off on that a little bit. Actual worthwhile effects of cannabidiol, beyond the realm of sort of withdrawal stuff that we were talking about, is that CBD seems to be perhaps, but potentially, one of the most important agents that we'll have in the future in terms of addressing panic. And panic is a really a tough area right now to treat pharmacologically. So that'll be one to really keep your eyes open about. So anyway, yeah, there's a lot of really great aspects of this particular topic. And I'm going to stop for just a second to see if Katie or others have things that they want to say, so that I don't just dominate the whole thing. But while people are doing that, there are some other things that I would like to address. So I'll stop for a moment.
Well, I just had a question for the students because I'm interested, not so much in the from the pharmacology perspective, but more like a policy perspective. How do you feel, and everybody feel free to contribute to this conversation. We're talking about this obviously from a very therapeutic approach and the fact that, you know, CBD could have all these positive effects. How do you feel about the fact that you can go into a grocery store or a gas station and essentially purchase CBD? You know, and the fact that when it comes to legalization, you know, people will be getting these possibly from a dispensary and not necessarily from a pharmacist. So I just want to see kind of how the group feels about something like that in terms of like execution and distribution. The fact that some of these obviously have generic drug names and will be prescribed through a physician, but there are a number of people who will probably, you know, purchase it for possibly recreational use or even self-prescribed. So I guess my question to the class is kind of, how do you feel about that as a future pharmacist?
I like the idea that regulation can make it safer because who knows what's being added. Because I mean, like, for one thing, I think like supplements have a lot of adulteration and stuff, and that is a little scary. And then you add this. I think having it available over the counter makes people think that there's less chances for interactions when that's just not true. Although there is something to be said for somebody being able to try to self-treat themselves and not have to like go into a doctor, especially when you have access issues, like a lot of people do. So I guess like the lack of regulation is a little scary, but at the same time, people have very little trust in healthcare these days and very little access. So it's a kind of a mixed bag.
Yeah, I think the accessibility issue is a great component to this as well, you know, and the access to healthcare and currently the cost associated with it and what's going to happen. I think it's such a hot topic, and you know, as Keith kind of alluded to, voting is so important, and as future pharmacists, it's going to affect you in so many different ways, from the regulatory standpoint and even from the healthcare standpoint, I think as well. And I'm getting some great messages privately as well. So thank you everyone for your thoughts, but please do feel free to share with the group as well.
I'll just jump in real quick and then stop again. And but the points that both of you are making there are excellent points, and that's a you know, a Pure Food and Drug Act sort of aspect to it, isn't it? Because the number one thing that the Pure Food and Drug Act of 1906 does for us is get a better handle on, as the name implies, the purity of a particular substance. And so where all that's going to play out with respect to cannabis is still anyone's guess. I just happened to see this morning, as I was looking at the things that come over the email sources that I have, discussion about the regulatory status of CBD as well as other cannabinoids. And you know, it's, as I think we all are aware, it really is a tremendously confusing area, because we've got the federal legislation, we've got each state having its own regulatory outlook on things, and then the interaction between the federal and the state governments, which is really complex because you know, take CBD as an example. It's regulated essentially both as a drug by FDA and as a food by the Department of Agriculture, and we have some, you know, differing things that are going on there. Is the original source of the CBD, you know, standard marijuana plants, or is it hemp? And that's a whole big area of confusion that's going on right now. So it's too bad that all of this is so confusing, because it's hard to, in a discussion like ours here, to land on a dime that says, you know, exactly the way things are. But hopefully with current legislation, pending legislation in Montana and other states, as well at the federal level, some of this will start to straighten out um, a little bit. But if we just go, to THC itself, um, at the federal level, marijuana is Schedule 1, uh, but THC itself has appeared on one of your slides, Katie, with the dronabinol, marinol. That's THC. That's a legal drug there, that can be prescribed and used medicinally, even though marijuana itself is Schedule 1. So for years there's been these tremendously dissonant sorts of things going on with legislation, and then when you get to the whole arguments, and you'll see some of this as you read the voter information pamphlet, where um, claims are made about really how little we know about cannabis. I agree with that in one sense, as we've already talked about, that there is more to learn, but I disagree with it in another sense, in that you could make a similar statement about just about any drug substance that you want, and so that's a difficult area to navigate and maneuver. Just in a historical context, of course, there's tremendous cultural information about marijuana that goes back thousands of years. And for example, cannabis has been recognized as a potential treatment of headache going way, way, way beyond the start of the scientific era. Then in the last few hundred years in the scientific era, there have not only been a lot of individual studies done, but huge commissions that have been brought forward by various governments throughout the world, including the United States, where there have the National Academy of Sciences, perhaps the most prestigious scientific body in the United States, has done extensive reviews of cannabis literature for many decades. There was a commission in Canada known as the Ledane Commission that issued results clear back in the 1970s on marijuana research, and the Indian Hemp Commission from the country India came out about 100 years ago with 7 volumes, I think, which would stack up thicker than the New York telephone book if we took a look at it in that sense. So while there's more to learn, there's also a lot that has been learned, and to interrupt regulation of marijuana by making claims that we just don't know much about it is to me not a valid sort of conclusion. One other thing along the regulatory sense that I would like to mention is that, and Katie, you emphasized the importance of the anticonvulsant properties of cannabidiol. That's a big deal. In the approval back there a year or two ago, as you mentioned, for specialized forms of epilepsy, was a huge breakthrough, and especially with respect to the treatment of childhood epilepsy, which has been extremely difficult to address pharmacologically. So anyway, I probably said a lot more than I was intending to there, but I'll back off and see what others might have to say or comment on this topic.
Yeah, I think the idea, the treatment for epilepsies are just so wide ranging. I mean, it's just, again, it's almost a description of a symptom, but there's so many different kind of physiological reasons that might be arising. So it's great when you see a possible therapeutic that can mitigate a lot of those physiological responses in the symptom of the epilepsy. Someone commented in the chat, Keith, and I'm just going to read it to you to see if you have any additional kind of thoughts, because I responded, but somebody was asking about the differences in lung function between inhaled marijuana use and the cigarettes, if there's if there's any comparison of the effects on lung function, and then do we know if marijuana is significantly less carcinogenic when smoked and inhaled? And my response to that was it's still not great for lung function, but then the carcinogenicity that we see with tobacco is often a byproduct of some of the additives that are put into cigarettes. And so I think that there's less carcinogenic effect, as I've read it, for marijuana use versus tobacco use. But I wanted to see if you had any additional thoughts or about that.
No, I think you've said it well. I think it is an area that still deserves further analysis. You know, with thinking about cannabis in general in terms of smoking it, there certainly are contrasting actions. There's bronchodilation actions which are beneficial. Then there's the potential, as you've been talking about, of carcinogenesis. My statements on that are often, as you burn anything, you've got potential trouble as far as carcinogenicity is concerned. But Katie, you make excellent points about the additives that occur in tobacco and tobacco itself. And tobacco smoke, as we all know, is tremendously complex in terms of the number of things that are in it, including, you know, straightforward carcinogens like the benzopyrenes to radioactive elements like polonium-210. And so, you know, smoke, be it tobacco smoke or cannabis smoke, is a complex mixture, and the name of the game changes anytime you start to burn anything. So those are certainly some things that are going to have to continue to be addressed in the future. One other thought that I had, Katie, you made mention there at the end of your presentation of the pharmacokinetic parameters, and these are a really big deal. The disposition of cannabis is a complicated one, and certainly, as you mentioned, with the tremendous lipid solubility of the cannabinoids or some of the terpenoids, for that matter, we're looking at some really long and drawn out releases of the metabolic products from the system. You know, one thing that I would predict as pharmacists that you'll encounter will be telephone calls that you'll get from people that say something sort of along these lines. This isn't a question that has to do with me, but one of my friends was asking me about how long can you detect cannabis metabolites in the system. I have to take a drug test for a job, and I would like to know whether some pot that I smoked a couple of weeks ago is going to show up in my blood test. And you know, with the tremendous sensitivity of detection methods anymore, trace amounts of cannabinoids can be detected for very long periods of time, weeks, or maybe even in the short month range. So it's a sort of a very different situation pharmacokinetically than you see with some other substances, which are eliminated, you know, so rapidly from the system within minutes or hours or a few short days. So I'm sort of jumping around and hitting a lot of different things here, but some of these things are popping into my mind as we go along. Okay, I'll stop again.
Yeah, well, Keith, we've essentially run out of time.
Yeah, and you know, before tomorrow, if some of these things rise to the surface and people want to talk about these things more, ask other questions or contribute thoughts that they have about it, there's no reason in the world why we can't start off our discussion tomorrow along these lines. I do want to say just one quick thing with respect to tomorrow's lecture. You know, we have more autonomic nervous system pharmacology to discuss beginning tomorrow. And so we'll start in on the sympathetic side. We finished up the parasympathetic side a few weeks ago. And what I would encourage people to do if they have time, is to look at that parasympathetic business. It's only 5 or 6 slides. And then be ready to think about that in the context of the sympathetic stuff, which will be our main topic of discussion tomorrow. Okay, sorry everybody for being so mouthy.
No, I think it's nice to have kind of just this open forum discussion about, especially about a topic that is so relevant right now. So again, if anybody has any questions or concerns, feel free to reach out to Keith or myself. Like I said, I'll be getting those exams back to you this afternoon and putting them in your mailboxes after you've looked those over. And if you have any questions, feel free to shoot an email to both Keith and myself, so we both kind of have, I think, a good understanding of what your question might be. And other than that, I think that's all we have for you today. And we're, I guess, looking forward to having you tomorrow. And Dr. Parker will be taking over at that point.
Thanks a lot, everybody. See you tomorrow morning.